I am S. Senthilrani, Assistant Professor, Department of Biochemistry, Seethalakshmi Ramaswamy College, Trichy. Now let us see what is metabolism. Metabolism is the process of converting food into energy through the process of digestion. Metabolism is also defined as the sum of all chemical reactions in the living cells that converts food into energy and building block. It is a series of chemical reactions that occurs in the body cells and tissues. Metabolism is also responsible for many vital functions such as breathing, digesting food, and regulating body temperature. There are two types of metabolic process. One is catabolism and the other is anabolism. Catabolism is a process mainly involved in breaking down large organic molecules into small molecules. This metabolic process releases energy. Anabolism is mainly involved in building up or synthesizing compounds from simpler substances required by the cells. This metabolic process requires and stores energy. In catabolism, there is breakdown of large molecules into smaller molecules and energy is released. The energy released is thus utilized by the small molecules to synthesize large molecules. The common metabolic steps involved: the simpler molecules enter into amphibolic pathway, that is either anabolism or catabolism. In anabolism, carbohydrates, proteins, lipids, nucleic acids are synthesized. In catabolism, carbon dioxide and water are produced. Metabolism of carbohydrates. Carbohydrate metabolism is a fundamental biochemical process that ensures a constant supply of energy to the living cells. It is a central metabolic pathway associated with the formation and breakdown of carbohydrate with energy generation. The most important carbohydrate is glucose, which is broken down via glycolysis, enters into the Krebs cycle and oxidative phosphorylation to generate ATP. The major metabolic pathway of carbohydrate metabolism are glycolysis, in which glucose is broken down to pyruvate, citric acid cycle, or it is known as TCA cycle. It is a common oxidative pathway for all the foodstuffs. Gluconeogenesis, synthesis of carbohydrate from non-carbohydrate source. Glycogenesis, synthesis of glycogen from glucose. HMP shunt, which is also known as pentose phosphate pathway. In this pathway, a 5-carbon compound, that is pentose, is produced, and also there is production of NADPH. In galactose metabolism, galactose is being converted to glucose 6-phosphate. Glycolysis. Glycolysis is defined as a sequence of reaction converting glucose to pyruvate or lactate with the production of ATP. This pathway is also referred as embedded Meyerhoff-Parnas pathway or EMP pathway. It is a universal pathway in the living cells. Now let us see about the salient features of glycolysis. Glycolysis takes place in all cells of the body. The enzymes for this pathway are present in the cytosomal fractions of the cell. It occurs in the absence of oxygen. It is called as anaerobic glycolysis. The end product is lactate. When glycolysis occurs in the presence of oxygen, it is called as aerobic glycolysis. The end product is pyruvate. Pyruvate is finally oxidized to carbon dioxide and water. It is a major pathway for ATP synthesis in tissues lacking mitochondria. Example, erythrocytes, cornea, lens, etc. So, glycolysis is very essential for the brain because brain depends on glucose for energy. Central metabolic pathway with many intermediates useful for the synthesis of amino acids and fat. Reversal of glycolysis results in the production of glucose, which is known as gluconeogenesis. Glycolysis mainly involves 10 steps. Now, let us see about the phases of glycolysis. There are 3 distinct phases of glycolysis. First, energy investment phase or priming stage or preparatory phase. The second phase is known as the splitting phase, and the third phase, energy generation phase or payoff phase. Steps involved in glycolysis. Now, let us see energy investment phase. Step 1, 2, and 3 are referred to as energy investment phase. In step 1, glucose is phosphorylated to glucose 6-phosphate by the enzyme hexokinase or glucokinase. Glucose 6-phosphate is more reactive than glucose. In step 2, glucose 6-phosphate undergoes isomerization to give fructose 6-phosphate by the enzyme phosphorexose isomerase. In step 3, a phosphate group is transferred from ATP to fructose 6-phosphate, producing fructose 6 1,6-bisphosphate. This step is catalyzed by enzyme phosphofructokinase, which can be regulated to speed up or slow down the glycolysis pathway. Splitting phase. Step 4 and 5 comes under splitting phase. Step 4, fructose 1,6-bisphosphate splits to form two 3-carbon sugars, namely dihydroxyacetone phosphate and glyceraldehyde 3-phosphate. They are isomers of each other, but only one glyceraldehyde 3-phosphate can directly continue through next steps of glycolysis. Step 5, dihydroxyacetone phosphate is converted into glyceraldehyde 3-phosphate. The two molecules exist in equilibrium, but the equilibrium is pulled strongly downwards as glyceraldehyde 3-phosphate is used up. Thus, all of the DAH is eventually converted. Energy generation phase. Step 6 to step 10 are called as the energy generation phase. In step 6, the two half reactions occur simultaneously. That is, glyceraldehyde 3-phosphate is oxidized and NAD plus is reduced to NADH and H plus. The overall reaction is exergonic, releasing energy that is then used to phosphorylate the molecule, forming 1,3-bisphosphoglycerate. In step 7, 1,3-bisphosphoglycerate donates one of its phosphate group to ADP, making a molecule of ATP and turning into 3-phosphoglycerate in the process. In step 8, 3-phosphoglycerate is converted into its isomer 2-phosphoglycerate. In step 9, 2-phosphoglycerate loses a molecule of water, becoming phosphoenol pyruvate. This phosphoenol pyruvate is an unstable molecule poised to lose its phosphate group in the final steps of glycolysis. In step 7, phosphoenol pyruvate readily donates its phosphate group to ADP, making a second molecule of ATP. As it loses its phosphate, PEP is converted to pyruvate, the end product of glycolysis. Substrate level phosphorylation occurs in step 10 and step 7. That is, a phosphate group is donated from the high energy intermediate substrate to ADP to form ATP. Significance of glycolysis, energy production. In glycolysis, glucose is broken down to pyruvate to produce energy. Energy backup. Glycolysis is vital reaction for all living cells. Red blood cells. Red blood cells which lack mitochondria. Glycolysis is the only source of energy. Muscle activity. Glycolysis enables muscle cells to generate ATP during severe exercise. Brain activity. Glycolysis break down glucose to pyruvate, releasing energy for the vital activity of the brain. Tumor growth. Increased glycolysis takes place in tumor cells. Therefore, that is a vital role for tumor growth and development. Thank you.